Good evening, ladies and gentlemen. Welcome to this ICT for Life Sciences Forum presentation and to the Melbourne Convention and Exhibition Centre, our first event here. So welcome. My name is Lewin. I'm the Executive Director of the Forum, and it's my great pleasure to welcome you here. There are a number of new faces in the audience I see, so perhaps for your benefit, to give you a 30-second overview about who we are and what we do. The Forum was established in 2008 really as an opportunity to bring together this important community of interest, people from biology, medical research, computing engineering, the physical sciences, what we now call convergence, the convergence research, transdisciplinary, some people call it. The general theme is to bring together scientists, researchers who have an interest in working together in multidisciplinary teams to tackle some challenging problems. And the purpose of the forum really is to provide the community an opportunity to hear from some of our leaders and to communicate to you some of the exciting things that are happening. So I hope you'll find this and other presentations, if you choose to join us in the future, worthwhile. It's my great pleasure and honour to invite someone to introduce our speaker this evening, someone who's probably one of the country's most respected and most loved researchers, scientists, and he's a great guy too. Please join with me in welcoming Sir Gus Nossal to introduce this evening's speaker.
Thanks very much, Lewin, and good evening, ladies and gentlemen. It's my very real pleasure to introduce a superstar of Australian infectious disease research and an extraordinary leader, Professor Sharon Lewin. Sharon is an infectious disease physician. She is the Director of Infectious Diseases at the Alfred Hospital. She's a Professor of Medicine at Monash University, and she's co-head of the Centre of Biomedical Research at the Burnet Institute. So she straddles teaching, research, and patient care in the most energetic and marvellous way. She's been president of the Australasian Society for HIV Medicine, and of course, as I think you all know, she's largely responsible for the great honour and privilege of Australia getting the AIDS Congress, which will be held in this very convention centre in July and going to be an absolutely crackerjack occasion. Her own research has ranged quite widely through the infectious diseases, but actually concentrates mainly on HIV. Its persistence, how and where and why it hides, and of course that in its turn leads her to think very hard about strategies for the cure of HIV. And that is the fantastic talk, ladies and gentlemen, that we're going to hear. Finding a cure for HIV, dream or reality. Please welcome Sharon Lewin.
Thanks very much, Gus, for the kind introduction, and thank you to Lewin as well for organising tonight. When I look out into the audience, I can certainly see a diverse group here. There's some very esteemed and experienced immunologists and virologists sitting up here, and some expert clinicians. I know some scientists from other disciplines, and I think perhaps some people that are not involved in science or medicine at all. So what I want to try and talk to you today about is a bit about the incredible excitement that exists now in the field of HIV, how far we've come, and then talk a little bit about the future of where we might go in whether we might one day have a cure, and some of the work that I do together with a team of scientists at the Alfred and Monash around that topic. So we have incredibly effective treatments for HIV in 2014, probably one of the greatest success stories, I would say, in modern medicine, where HIV now is no longer a death sentence, but a chronic, manageable disease, and often can be managed with a single tablet a day. On the left is what we used to use to treat patients in the mid-90s, when highly active antiretroviral therapy first became available in high-income countries. And on the right is what most of our patients are taking each day. A single tablet containing 3 anti-HIV medicines. Tablets now and medications now are non-toxic, have very few side effects, in fact are quite cheap and even available to many people in low and middle income countries. 11 million people taking antiretroviral therapy in low and middle income countries last year. At the same time, antiretroviral therapy also dramatically reduces a person's infectiousness. And this, we learnt this from an incredible study done of discordant couples, one partner positive, the other partner negative. And if we treated the HIV at the appropriate time, it reduced the likelihood of a negative partner becoming infected by 96%. So treatment not only makes an enormous difference to the person taking the medications, but also significantly reduces transmission, or what we now talk about as treatment as prevention. And as a consequence of this incredible advances in treatment, we are seeing dramatic reductions in HIV incidence globally. On the left is the number of new HIV infections reported since 2001, showing a clear decline, and in fact 50 countries last year reported a 25% decline in the number of new HIV infections. And on the right is the number of AIDS-related deaths, also showing a significant decline. And this is largely a result of treatment prolonging lives of the person taking the antiretroviral therapy, and treatment dramatically reducing infectiousness. However, you'll note that there are still 2.5 million new HIV infections occurring globally and close to 2 million deaths that still occur. So HIV is still with us, and HIV still remains a significant problem. However, with these dramatic advances of treatment, many of people have begun to talk about the end of AIDS, or really seeing the end of this epidemic. And there's one way currently that we think this might be possible, and this is through antiretroviral therapy. But the logistics of seeing an end of AIDS with therapy alone means finding, diagnosing, and treating everyone with HIV. And once you start treatment, you're on treatment lifelong. So the actual logistics of operationalising that plan, although we've done quite well, is really quite daunting. And that's why there remains this interest still now of finding a cure for HIV, or a way that people no longer need to take lifelong treatment. So what I'm going to talk to you now today is a little bit more about why we indeed need a cure for HIV. What are the major barriers to cure? Some of the potential strategies currently being tested, and then some current and future challenges in HIV cure research. So why do we need a cure for HIV? Well, I started by saying that antiretroviral therapy had dramatically changed the outlook for someone with HIV infection, which indeed is true. And this shows you the life expectancy of a large cohort of people in the UK with HIV in the dotted lines and without HIV in the solid lines. And you can see that if you treat someone with HIV and start treatment before the immune system gets too damaged, and we measure that by measuring someone's CD4 T cell count, which is normally at about 500. So if you start when the T cell counts reduce slightly to between 203 to 350, life expectancy now in both high-income countries such as the UK and Australia, low-income countries such as in Africa, is now the same as someone without HIV. But if you start treatment late, when there's evidence of immune damage, and you can see here in green when the CD4 counts between 100 to 200, or in brown when it's less than 100, when most people start treatment currently, globally, life expectancy still remains reduced. So antiretroviral therapy works very well if you start it at the right time. If it's delayed, life expectancy is reduced. We also know that there's still significant morbidity or disease that persists in people on antiretroviral therapy. And these diseases are largely diseases of accelerated ageing, an increased prevalence of cardiovascular disease, metabolic disorders, neurocognitive abnormalities, and a range of other end organ diseases that we commonly see in ageing. And this is largely due to low-level toxicity of the drugs, persistence at a low level of HIV, and something that we now know stays with people lifelong on antiretroviral therapy, which is low-level immune activation. And together they mean that there is still this ongoing problem of morbidity. And in fact, if an average day on the ward at the Alfred Hospital in 2014, many of our patients are admitted, their HIV's under control, and they'll be admitted for a range of these non-AIDS related diseases. And finally, the economics of long-term antiretroviral therapy is really becoming a great challenge for the global community. For every person starting antiretroviral therapy, there is one new infection, about 2.5 million of each. If we look at antiretroviral therapy coverage in low and middle income countries, it's around 40% of what it needs to be. We're treating about 40% of people who require treatment, so we're nowhere near the target of treating everyone who needs treatment. And the estimated costs of antiretroviral therapy and associated care is estimated to be $50 billion by 2030. So we need an alternative to long-term treatment. We need a way to see an end of AIDS, see an end to HIV, but without the bind of long-term antiretroviral therapy. So what I want to talk about now briefly is and try and explain some really complex concepts in HIV persistence. Why does HIV hang around in someone on treatment, and what are the major barriers to cure? So when you treat someone for HIV, we measure two things to see how treatment's working. We measure how much HIV is in the blood, or HIV RNA, and we measure their CD4 count, which is a measure of how good their immune system is functioning. When you start someone on antiretroviral therapy, virus very rapidly disappears from blood. In fact, within a month, a person can have from over 1 million copies per mil of virus in blood down to undetectable levels, or in most commercial assays, we can detect down to 50 copies per mil. And it will stay that way indefinitely as long as someone takes their treatment. And at the same time, immune systems recover in most patients, and someone with a very low T cell count, in most cases, will recover normal immune function. But as soon as you stop treatment, whether you've been on treatment for 3 years or 20 years, whether you started treatment shortly after infection or many years after infection, the virus comes straight back up. And in most people, it's within 2 to 3 weeks. So although this virus is so-called undetectable, it's always persistent and present. And we know that from a number of really important studies. And the first was to measure the number of infected cells. So again, for the people unfamiliar with HIV, HIV gets into its target cell, a CD4 T cell. HIV is an RNA virus. After it enters the cell, it copies itself backwards, what we call reverse transcription, and becomes DNA. And if you measure HIV DNA, which is another way of measuring infected cells rather than free virus, you can always find HIV DNA in people on treatment. So the virus is always there at extremely low levels, largely sitting inside a cell, and we can measure it as HIV DNA. And if actually you use a much more sensitive assay that measures down to 1 copy per mil, you can always find the virus there as well. But it sits there at very low levels of around 3 to 5 copies per mil, stay there indefinitely, no resistance, no rebound, just low-level persistent virus. So the $50 million question, of course, is where is this low-level RNA and low-level DNA coming from, and how could we eliminate those last bits of persistent virus? And we now have a very good idea about where the virus is hiding, and it hides primarily in 3 ways. The first is in what we call latently infected T cells, and I'm going to talk a little bit more about latency because that's the virus's key trick to persisting in people on treatment. Low-level viral replication, and then it has the capacity to hide in certain cells within anatomical reservoirs. So what do I mean by latency? Well, in a person who's on treatment and has this low-level HIV RNA, about 3 to 5 copies per mil, we find occasional infected cells which are latently infected. And latency is a very common strategy that many viruses use to evade the immune system and persist indefinitely in a host. And HIV is a master at it. And what HIV does is it manages to integrate into the host genome and become silent. The virus is still there, the virus can always emerge, infectious virus is still within this cell, but it is able to silence itself, and that's what we mean by latency. And even more brilliantly, it gets into a key infection-fighting cell, what's called a central memory T cell, designed specifically to last indefinitely because that's our immunological memory. There are cells that recognise foreign pathogens or new viruses, and we're armed with a library of different long-lived central memory T cells that persist for the life of a person. So HIV has this capacity to enter, integrate, and become silent in a long-lived central memory T cell. And these cells are really rare in a person infected on treatment, occur at a frequency of about 1 in a million. But we know infectious virus can always come out of these latently infected cells. The virus can be activated and release virus into the circulation. If the cell itself is stimulated, if it's responding to another antigen, like the common cold or flu, the activated T cell will release virus. But in a person on treatment, that virus can't go on to infect new cells. So while someone's on treatment, all of this stays under control, infrequent latently infected cells, low-level persistent viremia. And finally, we know that these cells can undergo low-level replication, or what's called homeostatic proliferation. So it's armed to persist there for life. The other way the virus can persist is by low-level viral replication. So antiretroviral therapy blocks most of the replication, but maybe not all of it. And we know this is the case in about 1/3 of people on antiretroviral therapy. But quite easy to manage because we can turn that off by adding in additional antiretroviral drugs. And finally, the virus can be sequestered in what's called anatomical reservoirs, the brain being extremely expert at harbouring long-lived infected cells, as is the gastrointestinal tract, the genitourinary tract and lymphoid tissue. So these are the major reasons why HIV persists in someone on treatment, and probably the most important reason is this ability to form these long-lived latently infected T cells. So given these quite formidable barriers, is dreaming or even contemplating or trying to find a cure even possible? And what are people aiming to do here? So ideally, we would love to cure HIV as we do all infectious diseases, meaning that we would need to eliminate all HIV-infected cells. If there were any HIV RNA would be less than what we could measure, or less than 1 copy per mil. And this is commonly now referred to as a sterilising cure, and we now know is really going to be very, very difficult. The other, perhaps more achievable goal would be to put HIV into remission, or think of it as we do a cancer model. So someone could have long-term health in the absence of antiretroviral therapy. The virus would be present, but at low levels, less than 50 copies per mil. And this is now commonly called a functional cure. And what we've learnt over the last few years is that a functional cure is indeed possible, and maybe something we could eventually achieve. And we know this from a range of case reports that have come out in the last 5 years, telling us that HIV cure is possible. And many of you may have read about these reports. The first to be reported was the Berlin patient. His name is Timothy Brown. He's an American man who was living in Berlin at the time, who needed a bone marrow transplant for the treatment of leukemia. and he received a bone marrow transplant from a donor who was naturally resistant to HIV. HIV needs one particular protein to enter a cell. It's called CCR5, and about 1% of people in this room will not express CCR5. They're otherwise completely normal, but they're CCR5 negative. So Timothy Brown received a bone marrow transplant from a donor who was CCR5 negative, and shortly after the transplant stopped his anti-HIV drugs, and the virus has never been found since that time in 2009. So here's the only example of sterilising cure. To date, hasn't yet been repeated. And it caused tremendous excitement because it was an illustration that perhaps cure is possible. Of course, cure could never be done through bone marrow transplantation. Highly toxic intervention, a death rate of about 25%. He, of course, had to have it for his leukemia, but it did cure his HIV. We then heard about the Mississippi baby about 2 years ago, and the Mississippi baby was a baby of an HIV-infected mother who was diagnosed at the time of delivery, and the baby was treated 30 hours after delivery. And 18 months, when the baby was 18 months old, the baby's anti-HIV medicines were stopped and the virus didn't rebound. The virus was still detected at very low levels, the DNA was still positive, but the virus never returned. So effectively, what I told you was called a functional cure. Again, we don't know what cured the Mississippi baby, whether it was extremely early treatment, whether there was something unique about an infant's immune system, meaning the virus could never get hold of those central memory T cells, we still don't know. And then finally, a group of patients from France called the Visconti patients, who were 14 patients who received very early treatment, stopped their treatment after 3 years. The virus remains clearly detectable at very low levels, and another 14 examples of functional cure. So as a doctor, we're always trained to, not take too much notice of the anecdote, an unusual case. What really does it mean for the 35 million people infected with HIV? Well, what these cases have done is given people hope, certainly given patients hope that a cure is possible. They've given scientists hope that a cure is possible, and they've each influenced many of the ideas and much of the science that's being now tested to see if we can mimic these cases in other ways. And I want to talk about some of those approaches, including some of the approaches we've been testing here in Melbourne. So what are some of the ways that we could eliminate latently infected cells? Well, we could treat during acute infection or treat very early, a la the Mississippi baby or the Visconti patients. We could activate latent infection. I'm going to talk a little bit more about that approach. We can boost HIV-specific immunity, which is important for keeping HIV under control. And maybe allogeneic stem cell transplantation or bone marrow transplantation could work in other settings. So how would treating acute HIV infection work? It would mean very early antiretroviral therapy. We usually wait until there's signs of immune damage before treatment. But if we treated as soon as someone was infected, this perhaps could limit the number of latently infected cells. It would prevent this phenomenon of chronic immune activation that we know can fuel ongoing viral replication. It will preserve HIV-specific immunity, and it might achieve a functional cure. And there's now many reports of treating very early antiretroviral therapy. In fact, an amazing study done in Thailand where they were able to treat people within 15 days of infection. And indeed, it certainly limits the number of latently infected cells. But functional cure is still rare, even with very early treatment, and probably occurs in about 5 to 15% of people. But we need to understand better why those 5 to 15% of people are able to keep their virus under control with the help of early antiretroviral therapy. So probably a most promising strategy for seeing a cure for HIV, but one that's really not that relevant to the majority of people infected with HIV. Most people don't know when they become infected with HIV. only 50% will have a glandular fever-like illness, and 50% will be completely asymptomatic and never know they contacted the virus. But still an area of intense research. The other approach is one that I have worked on for quite some time, and the idea is to actually wake up or activate latent HIV. And the principle here is a little bit counterintuitive, but I've already explained to you that latency means that virus as HIV DNA, buried as part of the host DNA, sitting inside a resting cell. And the idea is that if we could activate that virus so that the virus started to make copies of itself, which is making RNA, started to make proteins, which would then become visible to the immune system, or started to actually produce virions, usually when a virus leaves a cell, the cell self-destructs. So if you could effectively wake up the virus and push it out, this could lead to virus-induced cytolysis or activation of an immune response to recognise this recently awoken cell. And this would be done in people that are on antiretroviral therapy, so that if we made any new virus, in the presence of treatment, it couldn't go on to infect new cells. And we now know there are many, many compounds that can do this in laboratory models of latency. And we've developed many models that we test and screen different drugs to see which ones can effectively wake up latent HIV. And every week there's new compounds that are described that can activate latent HIV. I've listed some of them here, and the ones that we've been most interested in are these drugs called histone deacetylase inhibitors, or HDAC inhibitors. And they're of great interest to us because these drugs are already licensed for the treatment of rare forms of skin cancer, cutaneous T-cell lymphoma, and therefore were drugs that we thought we could potentially move into the clinic quite easily. There are other drugs that do it, drugs such as cytokines that wake up the immune system, one in particular, IL-7. Disulfiram, which the drug antabuse used to treat alcoholism, also can wake up latent HIV, some rare antibiotic classes, and then a whole group of compounds that are largely involved in regulating gene transcription, or what we call epigenetics. And these compounds are very relevant for HIV because HIV basically behaves like a host gene. Epigenetics means control of gene expression or gene regulation, and we know a lot about epigenetics largely through cancer research. And HIV essentially acts like a host gene, and so we can take advantage of some of that knowledge to manipulate how HIV is expressed. So if you think about HIV as a gene, in a latently infected cell, it is a silent gene, which basically means that the chromatin, shown here in grey and orange, is tightly coiled. The histones, which the DNA wraps around, is silenced or not acetylated, and basically that gene or HIV is turned off. This is what HIV looks like in a silenced, latently infected cell. And in an actively replicating cell, HIV is turned on, the chromatin is open, the histones are acetylated, and the relevant bits and pieces needed to drive copying of the HIV virus are all in place. And we know that these drugs, histone deacetylase inhibitors, can actually transfer HIV from this state, the silent state, to an actively replicating state. Double negative histone deacetylase inhibitors, they'll acetylate the histones. So we became very interested in these compounds several years ago, back in 2009, and we're following the development of these compounds largely in the oncology arena as we got better and better and more potent HDAC inhibitors. So back in 2010, we started a study of patients with HIV on treatment, on antiretroviral therapy, been on treatment for 3 years, their virus was at low levels, less than 50 copies, they had normal T-cell count. And we gave them this drug called vorinostat, which is the first HDAC inhibitor that was licensed for the treatment of cutaneous T-cell lymphoma. And we gave them 14 days of vorinostat and asked a pretty simple question at the beginning. It's got more complicated since as the field has moved on rapidly, but the simple question was, could the HDAC inhibitor or vorinostat wake up latent HIV, force it to start expressing itself after 14 days of administration? And so we did this by measuring RNA inside the cell. And indeed, we found that it really did wake up latent HIV, because when we measured RNA inside the cell, cell-associated RNA, that shows you the fold change in RNA for the 20 patients enrolled in the study. The maximum fold change in some patients was really high, about 20 to 30-fold increase in cell-associated RNA. And in some patients, minimal response. But overall, about a 7-fold increase in cell-associated RNA. So this study, together with another similar study done in the US where they gave a single dose of vorinostat, really challenged this idea that latency is a permanent state of silenced virus. Did waking up the virus kill the cells? Well, after 14 days, we unfortunately found no change in HIV DNA or RNA. So we measured virus in the plasma using a very sensitive assay that measures down to 0.3 copies per mil. We saw no change over the course of the study. And when we measured HIV DNA, we saw also no loss of infected cells. So we were excited to see that HIV RNA could be produced, but we still have no evidence that we had loss of infected cells. Now, vorinostat was really the first medication off the rank, the first of its type that was licensed. And since then, other HDAC inhibitors have been investigated. This is work that we did in collaboration with a group in Denmark, which gave another HDAC inhibitor, a more active HDAC inhibitor called panabinostat. Shown here, was given in 4 cycles, shown here in yellow. And they found very similar changes, meaning that the RNA inside the cell increased each time a course of panabinostat was given. So again, this drug was able to wake up that latent virus. This time there was evidence of virus production in plasma, but again, no change in DNA. One issue with this approach of activating latent HIV is that all of the tools that we've got at the moment are not specific for HIV. So HDAC inhibitors are a very blunt instrument that will turn on thousands of genes in addition to HIV. So latent infected cells are rare, only 1 in a million, and we've been giving medications that are going to have very significant effects on many uninfected cells, and that's a real challenge at the moment. We have no HIV-specific drugs to wake up the virus. And we actually looked at this using a technique where we can look at thousands of genes at the same time. And this is a complicated slide. It's called a heat map, where we look at genes that are turned on, increase expression, and it shows up as red, or genes that are dampened or decrease expression, it's shown in blue. And this technique allows you to look at thousands of genes at the same time, and each row here is a different gene, and each column is a different sample from 9 patients that participated in our study who had samples collected at baseline, shown here in green, and the same patients who had samples collected at day 84, 2 months after stopping vorinostat. And to our surprise, we saw very marked differences in gene expression at baseline compared to months after receiving the drug. So the names of these genes are of high interest to us, of less interest to you, but the point is that we significantly altered a very large range of host genes with just 14 days of drug. And this is a big issue in patients who are doing pretty well on 1 tablet a day. So we need to balance the enthusiasm around finding agents to activate HIV and what could potentially be significant safety issues. We don't know what this means, of course, and we're following our patients currently in a long-term follow-up study. So one big question now is we have a lot of agents that can activate HIV transcription. The question is whether that really is going to lead to cell death, and to date we have no evidence of it. What might be a strategy that will be pursued is that we need to both activate and then kill the recently woken cell with something like a therapeutic vaccine or a way to pump up the immune system. And there's a range of new molecules that can do that, one of those being a therapy that targets something called PD-1. Finally, transplantation. I told you a little bit about the Berlin patient and a very exciting case report. As I mentioned, he was HIV positive, he had leukemia, he had normal expression of the molecule CCR5, or what we call CCR5 wild type. As part of his transplantation, he received chemotherapy, total body irradiation. He then received a bone marrow transplant from this CCR5 negative donor. He got something called graft versus host disease, which is when your graft, your new bone marrow, reacts against your own cells, and that also may have assisted in wiping out any latently infected cells. And he now is 5 years post-transplant and stays cured. But we really don't know what cured Timothy Brown. Was it the graft versus host disease, the chemotherapy, the fact that he had a CCR5 negative donor, or just by receiving a bone marrow transplant? So several years ago, there were 2 more patients reported. These are called the Boston patients. They were HIV positive, on treatment. They had lymphoma, and they received reduced intensity irradiation. And they received a bone marrow transplant, but this time from a CCR5 wild-type donor. So the donor was susceptible to HIV. And there was a lot of excitement when the physicians caring for these patients noted that at 6 months, once the transplanted cells had taken hold and populated the bone marrow, these patients were now HIV DNA negative and HIV RNA negative. And it raised the possibility that maybe you didn't need this fancy bone marrow that was CCR5 negative. Maybe transplant alone could have eliminated latently infected cells. So the proof of whether these patients had been cured is, of course, would be told once they stopped treatment. So the first patient did stop his treatment. It was only just last year. The patient remained HIV negative. His DNA was negative. When they measured RNA in blood, that remained negative for 12 weeks, which is quite a long time because most people rebound in 2 to 3 weeks. And then over 2 days, the virus just took off again. So you really don't need much virus to set the whole thing going again. So he went from being completely DNA negative to over 1 million copies per mil in a space of about, initially this initial period was over 2 to 3 days. And similarly, the second patient got about 32 weeks virus-free, again, a very long time, so that the transplantation had really reduced the amount of residual virus, but it taught us that maybe only, there's some nice mathematical modelling thinking how many infected cells were needed to reactivate by 32 weeks, and the estimates are around 200 infected cells. But look at this, from undetectable to over 1 million copies within 2 days. And what might be unique about these patients is, of course, they have a totally new bone marrow, a bone marrow that's never seen HIV before, totally naive immune system, so a little bit of virus can just take off very quickly. And it's really raised a big question in the field of how low do you need to go? Is it possible really to eliminate every infected cell? And I doubt that really is possible. You're going to need a combination of few latently infected cells and some level of immune surveillance to keep it under control. And finally, a really exciting approach to maybe curing HIV, again inspired by Timothy Brown, is making cells resistant to HIV. And this largely comes through gene therapy, or the capacity to eliminate a gene of interest using literally gene scissors, much smaller than the ones that you see here, but they do exactly the same job, recognise the gene of interest, and we can do this for CCR5. So make someone's T cells CCR5 negative, so therefore resistant to HIV, and you actually can use this same approach to chop out HIV that's sitting inside the integrated latent reservoir. So this is how these studies are already being done, some completed, all in the US at the moment, where a person with HIV on treatment undergoes a large bleed or what's called leukapheresis. The CD4 T cells are a mix of CCR5 positive or HIV susceptible and CCR5 negative. Ex vivo, those T cells are modified using these gene scissors or what's called zinc finger nucleases to eliminate CCR5. The CCR5 negative cells are expanded and then reinfused back into the patient. And there's been several trials now already completed. The first was reported in the New England Journal a couple of months ago, and it showed that this is achievable, it's safe, and that the CCR5 negative cells persisted in the patients at a level of about 6%. So of the 12 patients that were in this study, 6 then stopped their treatment. In all 6, the virus came back. But this is what happened to their T cells. So after infusion, you can see this increase in all of their T cells, the unmodified cells in blue, and their CCR5 modified cells in red. So about 6% of the cells were now CCR5 modified or resistant. These patients underwent a treatment interruption, stopped their virus comes back rapidly, and when virus replicates, it knocks off CD4 T cells. And what was encouraging about this was that you can see the unmodified CD4 T cells die very quickly or decline very quickly, while the CCR5 modified T cells hang around longer or have a longer half-life. So there was definitely a survival advantage of these CCR5 modified T cells. To keep the virus under control, we probably need much higher levels than just 6%. A lot of work now being done on ways to increase the number of modified T cells that can be transplanted. So there has certainly been a lot of advances and interest and development in the areas of thinking about how we might cure HIV, but there's many more challenges to face. I think a big challenge is balancing what's acceptable risks and toxicities in a population doing very well on treatment. These are not patients with malignancy or leukemia or facing life-threatening illnesses. These are patients that certainly want to be cured from HIV. We have had absolutely no problem in enrolling patients into our studies, but we really need to think very careful about acceptable risks and toxicities. There's a lot of discussion on the ethical and regulatory issues. There's a big challenge about managing expectations of both the study participants and the community. There's certainly, I started by saying how these cases have given many people hope, but we don't want to replace that with hype. Curing HIV really remains a very significant scientific challenge, and we're really only at the very first stages of addressing that. I think we still need much more greater collaboration and greater engagement of the private sector in the effort to date. There hasn't been much involvement in industry in the efforts. And I think any talk about cure can't get in the way of what we know works now for HIV, and that's access to antiretroviral therapy. 40% of people in low-income countries on treatment, but we really need to work hard as a global community to get that up to 100%. And there's a lot of efforts going on. I thought I'd just briefly mention some of the work that the International AIDS Society is doing in really driving the agenda here and enhancing and advocating for more funding and collaborations and involvement of industry, involvement of the community, a great effort being driven by the International AIDS Society. There's no doubt there's also an really significant increase in funding towards cure research, and I hope together, because I don't think money alone is going to crack a really big, important question in science, but money together with these other initiatives could really see the field accelerate in coming years. So in conclusion, I've shown you multiple but rare examples of cure, and that's given hope that cure might be achievable. There are many strategies being tested, but all of these are really early proof of concept studies. I think it's unlikely that we're going to achieve a cure just by reducing the size of the reservoir. You really don't need many viruses to set the whole thing off again, and enhancing immune control will be needed. And I think very prevalent for all of us is that whatever is discovered, there's going to be a significant additional challenge to find a strategy that ultimately is cheap, scalable and widely available to people who need it. So I just want to acknowledge a large number of people whom I work with, particularly Julian Elliott from the Alfred Hospital, who led the study on vorinostat. Many members in my lab at Monash, particularly Fiona Whiteman and Ajantha Solomon, who did a lot of the virology and immunology on the vorinostat study. We work closely with investigators at the Peter MacCallum Cancer Institute, Miles Prince, who have been expert in these drugs and the management of cancer, as well as the National Association of People Living with HIV. And funders, including the Australian Government, National Health and Medical Research Council, the National Institutes of Health, through a fantastic private-public collaboration which we have with UCSF and Merck called DARE, and the American Foundation for AIDS Research. And before ending, Gus mentioned earlier about the International AIDS Conference. We're really excited to be hosting the conference here in Melbourne in July. I'm co-hosting the event with Françoise Barré-Sinoussi, the woman that discovered HIV in 1983 and won the Nobel Prize. And we're expecting 14,000 people at the event. I think what's particularly interesting, especially for those of you in the audience who are non-scientists or not clinicians, is this is not a regular scientific conference. It's really much more than that. The conference over the years has really galvanised great changes in the response to the HIV epidemic, and that's because it's not just a scientific program. There's a huge representation here by affected communities, a very strong representation by leaders and people that can really make a difference, and funders, etc. So we're hoping that it will really take over the city in many ways, and there's many ways that you can be involved. You can, of course, register and come. You can participate in over 120 affiliated events that will be taking place in Melbourne. There will be a daily TV show, you can watch it online. There are parts of the conference which are available and open and free to the community, particularly the Global Village, an incredibly vibrant space that will host cultural activities, performances, community and art exhibitions, which will be in the exhibition centre. And lots of ways to find out what's happening through social media. And hopefully, as a person living in Melbourne, this will be a great opportunity for you to get involved. So thanks very much. Thank you.
Thank you very much indeed, Sharon, for a really beautifully clear, wonderfully delivered talk, magnificent slides. Couldn't have possibly wished for anything happier, and Lewin is already smiling all the way. You mentioned the brain as a reservoir. Now, with whatever means you're going to use to flush out the virus and then polish it off, drugs, antibodies, whatever, is the blood-brain barrier going to be a problem for you?
Yeah, it could be. So in the brain there are long-lived specialised cells that can be infected, like astrocytes and microglia, and there's also that barrier that limits penetration of a good immune response. And I personally think there's evidence that the brain is a problematic reservoir in some patients, but not all. And probably a good example of it not being a major problem is the Berlin patient, or where obviously there was no specific treatments aimed at these long-lived infected cells in brain, but it wasn't a source of rebound. In fact, the Berlin patient had a brain biopsy for another reason, no evidence of virus there. So I think it could be a problem in some patients and certainly needs to be addressed, but maybe not in all patients.
Sharon, thank you for a fantastic and fascinating talk. Can you tell me what the population modelling says about what proportion of cases would need to be treated with current therapy in order to stop transmission over a generation or two by bringing the R naught below 1?
Yeah, that's a great question, and there's been a lot of really good modelling about that, and not just good modelling, actual real evidence of it. So there's two fantastic papers published in Science late last year showing that in parts of South Africa, if you could just reach a level of 40% of uptake of antiretroviral therapy, there were already significant declines in incidence of HIV. So you probably don't need to treat every single person to block transmission, but it's already been shown that if you get up to 40%, and that's been shown in other settings such as injecting drug users in British Columbia, etc., to actually eliminate HIV, all transmission, it probably needs to get a lot higher than that. And of course, we're still left with the problem of 35 million people already infected. But there's some really nice modelling, and I think there still is, and I wanted to try and really convey that to you, that we really know now that treatment, and if we could really deliver treatment to a large number of people, we can already now and already have had a major impact on the epidemic.
Professor Lewin, you made a really good case for starting HIV anti-retroviral therapy pretty much immediately. What would that do for your risk for some of the morbidities that you spoke of, often to do with the medication?
Yeah, it's an area that's quite hotly debated, actually, when to start treatment. And for years, we've always waited till we see a bit of immune damage. And in fact, in Australia, we could only prescribe drugs when there was some evidence of immune damage or when the CD4 count's less than 500. But many countries, including France, Thailand, the US, have moved to this model of just as soon as you're positive, start treatment, what's called test and treat. So they prescribe at any CD4 count. And in fact, our reimbursement system just changed in April, so we can do the same here. But we actually don't know whether there's clinical benefit. We know if you treat we know if you wait too long, your risk of toxicities and morbidities is higher. We don't know if you treat above 500 whether we reduce that completely. And there's actually a big randomised study underway trying to answer that question. The rationale more for very early treatment is probably only save 2 or 3 years by waiting, and you could reduce transmission. And that's what's pushed many countries to this test and treat model.
Hi Sharon, thanks for the great talk. You might be aware that there are individuals whose CD4 immune system is able to produce widely or broadly neutralizing antibodies against HIV. What do you think of the chances of actually promoting that in individuals who don't have that natural ability?
Most people do a pretty good job at keeping HIV under control by either making antibodies, as you mentioned, or making effective T cells. But no one can actually eliminate the virus, but many patients can keep it under control. And there's been a big debate about what you need, antibodies or T cells. And as you say, recently there's been this description of some people that make really effective antibodies, broad, meaning they've recognised lots of different viruses, and neutralising, meaning they can sort of stop the virus in its track. But what we know, if you make broadly neutralising antibodies, you often make them too late. The virus is already 3 steps ahead of you by the time you make the broadly neutral. Those lucky people that make them don't get any benefit themselves from making those broadly neutralising antibodies, because by the time the immune system's made them, the virus is already 3 steps ahead. But finding those rare individuals with broadly neutralising antibodies has allowed scientists to take them and manufacture those same antibodies in the laboratory. So in fact, that's one area of vaccine research that's really accelerated in the last 12 or 18 months, the capacity to synthesise those broadly neutralising antibodies. So to date, we've never had an antibody vaccine that's worked for HIV. Broadly neutralising antibodies haven't yet been tested as a vaccine, but I think they certainly one of the the big promising advances in the last 18 months. What role they have in cure research, less clear, because you need to kill that actual cell, not just stop the virus in its tracks. But maybe a broadly neutralising antibody can trap any virus that's released from a latently infected cell. But there have been big advances in manufacturing those antibodies in laboratory models.
Well, passive antibodies gave von Behring the first Nobel Prize ever in 1901 for diphtheria. Who knows?
Who knows?
It remains only, ladies and gentlemen, to state categorically that the invited superstars such as Françoise Barré-Sinoussi, Tony Fauci and Peter Doherty had better look out, because this talk is going to sideline them. Thank you all very much. Let's show our appreciation.
In conclusion, I'd like to obviously thank you all for being here this evening, and to thank in particular the Melbourne Convention and Exhibition Centre for hosting us, and Susanna Bishop, I beg your pardon, the General Manager of the Club Melbourne Ambassadors Program in particular, who provided the support to make this event here possible. So thank you very much, Susanna. And I guess the Club Melbourne Ambassadors Program, for those of you that don't know, is an initiative with the Melbourne Convention and Exhibition Centre, works with academics such as Sharon, who's an ambassador at the club, to bring events such as AIDS 2014 to Melbourne. So it's a really important economic driver for the city. So Susanna and her team do a fantastic job, and our ambassadors like Sharon, Lewin do a terrific job in working for... how many years did you work to get this to Melbourne, Sharon? A couple of years. So it's a significant effort, and as you've been told by Sharon, significant activities around the conference. It's going to be a very significant event for the city. I'd like to thank our sponsors, without whom none of this would be possible, so they're all listed up there. I thank them each for their ongoing support of the ICT for Life Sciences Forum. Can I please ask you to join me in thanking Sharon for her commitment to be with us this evening and share some of the exciting things that are happening in the field, and to also wish her well with AIDS 2014, and in your new capacity as the founding director of the Peter Doherty Institute. We wish you and your team over the many years great success. And I guess I couldn't miss the opportunity to also ask you to join with me in wishing Gus a happy birthday in about a week and a bit, your 83rd birthday, Gus. So we'll round off with a couple of thank yous. Have a safe journey home, and we'll see you next time. Thank you very much.